My name is Paul Gebz, professor at the University of California, Davis. I will give you a talk about the main reason we are here, and that is this ABC project, the African Bean Consortium project. I'll explain to you what the general goal is, what some of the characteristics are, and before that, I would like to try to put this project in a broader context of agriculture, food, and so on. So I'll talk briefly at the beginning, challenges in crop improvement, also opportunities in crop improvement, what is the goal and organization of the ABC project, specific goals, specific guidelines that we follow, what is the status currently of the project, some of our concerns, and ultimately where do we want to achieve, where do we want to go, and what are the criteria to determine if we have been successful or not. So I think the challenges in crop improvement, one of the main ones is that we have to meet the demand. Will we be able to meet the demand with sufficient supply of food? Okay. I'll remind you that one of the Millennium Development Goals is precisely eradicate poverty and hunger. And I think that it's interesting that the two are linked in the same goal, poverty and hunger. There has been broad progress on several of the Millennium Development Goals. I'll have listed here a few, having the poverty rate, having population without access to clean water, and so on. But hunger so far, only limited progress has been made. Okay. There are still about close to a billion people who are undernourished, means about 15%, and child undernutrition, which is really a serious problem. It is still a major issue. If that were not enough, I think, is that we are now faced with global climate change, and we can expect almost certainly, I would say, a 2-degree centigrade increase in temperature, if not a 4-degree. Okay, so, and it has been estimated that that is going to lead to a reduction in agricultural production with the current technology, if we don't do anything else. And for Africa, it would be about 10 to 20%. One of the Millennium Development Goals in general is to eradicate this extreme poverty and hunger. And we have to put this in the context that currently we are at 7 billion people, and this is going to increase to 9 billion people almost certainly. We can discuss what's going to happen after the 9 billion. Is it going to flatten out or not? And you see that extreme poverty is falling in almost all of the regions, but it's fairly slow. Okay. Another way to look at it, which is very important, is you see that the number of malnourished people is actually decreasing in relative terms. But in absolute numbers, it's still constant. That is because we are still in this mode of population increase at the level of the entire planet. We see that progress in relieving food deprivation has slowed or stalled in many regions. So we are still faced with this potential imbalance between supply and demand in agriculture. To make things even slightly more difficult, is that you see here that countries in sub-Saharan Africa have been the hardest hit by the food and financial crisis recently. Since 2008, you see this kind of increase in the number of undernourished people. It has increased recently in Africa, whereas in other regions, like in Asia, for example, the decrease has stopped, but it's not increasing. Okay? And so nearly 1 in 5 of the children under age 5 in the developing world is underweight. And that has long-term consequences also, actually. So climate change is that second major factor that is appearing, and you see that aside from some regions at high latitude, we see that may see an increase in agricultural production. Most of the other areas will suffer from a reduction in agricultural production. And that includes even regions in temperate regions, like in the US, for example. We can look here. This is a study that was done on the potential production evolution of both maize and beans, two important crops. And in this particular region here, you see Kenya, Tanzania. This is for maize, this is for beans. I'm not a specialist in these predictions, but what strikes me is that we may dispute the actual precision of these estimates, these predictions. But what it also shows is that there may be regions in which agricultural production will increase, the green areas, and other regions where that production is going to decrease. And it tells us that we have to be very attentive in terms of local adaptation of crops. Okay? That is, how that is going to evolve, we don't know for sure, but we have to set in place breeding systems that will take into account the local changes in climate, for example. Okay? Now, the emphasis then is on trying to adjust agricultural production and make it, what they say, climate change resistant. And most of the emphasis has been placed on cereals, but it's clear also that we need kind of a broad type of diet, and so we have to address legumes and other crops in addition to cereals. And actually, the evolution of agriculture presents us with an example that I've listed here, a number of centers of agricultural origins, and each of those you see a combination of carbohydrates and protein crops. So you take, for example, in Mesoamerica, we talked about maize and beans. It comes from Mesoamerica. You see combination corn and as a major source of carbohydrates, and phaseolus beans as a major source of protein, plant protein. You see Africa here, you have a series of cereals, African rice, pearl millet, sorghum, teff, fonio, and also a series of legumes, for example, native African legumes. The same thing holds for China, Southeast Asia, for example. The Andes is somewhat of an exception in that the source of carbohydrates there are primarily root crops. Originally at least, and then later joined by corn, and then you have a series of protein crops, peanut, phaseolus bean, jack bean, lupins, and so on. Okay? So people, even without having a scientific background, have adjusted and have focused on having a diverse diet. Okay? So this project here emphasizes legumes particularly, and legumes do play an important role in alleviation of hunger and malnutrition, this first Millennium Development Goal. They help reduce rural poverty because you have different possibilities of income generation, not only seeds, but also pods, leaves, oils, and so on. It is an example of crop diversification. In California, where I'm from, legumes play an important role in rotations, for example. But it's also, you have cases of double cropping, multiple cropping, relay cropping, all involving legumes and other crops. There could be cereals, there could be solanaceae, and so on. They complement cereals not only agronomically as a source of nitrogen, but also nutritionally. They also play a role environmentally. And again, in California, there is a serious concern about the use of nitrogen fertilizer, which contaminate groundwater. And one of the proposals is actually to increase emphasis on legumes, not only as a grain crop, but also as a cover crop, for example. Legumes have high positive income elasticity, which means that when income increases, the consumption of legumes also increase, and more so than for cereals. Poorest countries derive more dietary proteins from grain legumes, and I've listed a few examples here in Africa, where it can go from anywhere between 15 to 40%. Okay? And the production so far of grain legumes has outstripped population growth so far. Okay? Nevertheless, there are some issues with grain legumes, and in general, the increase in production of legumes has lagged behind those of cereals. Okay? It's estimated that the yields of legumes need to increase faster than the current rate, about 50%. And you see that the current rate of increase of legumes is about 0.4%, and compared to cereals, which has been 1.5%. The causes for this low yield growth, I think, are multiple ones. Generally, legumes receive fewer inputs than cereals. There is also a push, legumes get pushed into marginal areas. The major producer, for example, of legumes, of beans in the world is Brazil, and beans have been pushed away from major production areas by other crops. And so the soils are less fertile, there are other pests, and so on, actually. Okay? There is also less policy support than other commodities, which receive subsidies, for example. And also, in general, and this is where this ABC project comes into focus, is that there is limited research and development and spread dissemination of improved technologies, like improved varieties, for example. Okay? And so, a lot of the research that has been done in legume focuses primarily on production constraints, such as tolerance to biotic or abiotic stresses. Okay? And in this project, the ABC, we focus primarily on biotic stresses, so resistance to diseases, primarily. Now, what about the opportunities in crop improvement? What can we do to accelerate this pace, hopefully accelerate this pace of yield increases? I think that the progress primarily in genetics and genomics and genetic engineering, combined with traditional breeding. Okay? In genetics, linkage mapping has been a source of information for genes controlling agronomic and consumer traits. We know where they are, we can estimate what's the magnitude of their effect. In genomics, we can actually get information about plant function. But also, it is a source of sequences for marker-assisted selection, which is also, again, one of the major foci of the ABC project. And then something I will not talk further about, but genetic engineering also can play a role for traits for which there is limited genetic diversity in the crop of interest. Okay? So this project actually intends to speed up the availability of genetic and genomic information in breeding programs. And especially for East African bean breeding programs. So why focus on common bean? It is because it's culturally quite important in East Africa. Okay? The interesting thing is that it's not a native legume, but maize also is important, and it's not a native cereal. And it's almost like the entire cropping system, which was devised in the Americas, has been imported into East Africa and has superseded the native cropping system. And the other reason for the focus on common bean is that it's currently among all the dry beans for human consumption, it's by far the most important one. So culturally, it is quite important. They also have a nutritional role. They are a source of protein. I've listed some of the countries, and you see that 20 to 40% of the diet intake in protein comes actually from beans. In addition to proteins, you also have minerals, vitamins, fibers, and so on that play an important role. And you look at common bean, they are used primarily for grain, but also pods, that's a source of revenue, also leaves, and in some cases they actually also eat the flowers in salad because they are sweet, and it's the nectar produced by flowers. In East Africa, common bean is very important. You see here, this is for dry bean, and you see here several countries. You see Tanzania, Uganda, Kenya, and then there is also Rwanda here, are major producers at the world level. For green beans, you don't see the same picture, but I think that is about to change. That is going to change because green beans are a source of cash for export. Okay? So genetically speaking, the two are fairly similar. So the ABC project here is funded by the Kirkhouse Trust, whose main goal is to support research in agricultural crop improvement for the developing world, primarily Africa and South Asia, and specifically for legumes also. Okay? In this case, it's to introduce the capability of conducting marker-assisted selection, I'll come back to that, and integrate that into the regular bean breeding programs, phenotypic selection mainly. The general organization is that there is a number of sub-projects. There are African country projects which are affiliated with universities or agricultural research organizations, depending on the country. There is a US project in which I'm involved, which looks at the development and support of this marker-assisted selection, and I'll say a few more words about that. We also have assistance from CIAT, which is the International Center for Agriculture in the Tropics, actually, which is located in Colombia, but it has an important lab in Uganda. Okay? And they have been helpful in short-term training in marker technology as well as plant pathology techniques. And we're also considering they may also play an important role in the future in terms of comparative information on pathogen variability and disease inheritance. Okay? And then there is also an important component. These are the administrative and technical offices of Kirkhouse Trust in the UK. And so all of these projects work together to achieve this end goal. Some additional considerations are that the project favors education and research in Africa, mostly. That it, in addition to the research, it wants to improve the human and physical infrastructure, hence training, hence also the development of simple but effective labs. Something that is dear to my heart is that we want to focus on researchers involved in the group becoming independent researchers capable of conducting marker-assisted selection. And I'll come back to that later on. The status of the project is that we are about halfway to the first phase, both in terms of the African projects developing improved advanced lines, as well as the bioinformatics in the US to support the marker-assisted selection. So I've told you that we are focusing for this project mostly on diseases. And we have identified with participants 5 major diseases. And in turn, each country is focusing on 2 or 3 major diseases. So these diseases are, in the case, there is a virus, bean common mosaic virus and bean common mosaic necrosis virus, and they're related. The distribution you'll see of these diseases is worldwide, and that is in part because they are seed transmitted. So when people exchange seeds, they also exchange pathogens, for example. They are seed propagated, in this case also vectored by aphids, and there is resistance available, has been identified, and you see a number of the genes here. In turn, these genes have already markers that tag the resistances. Another one is a bacterial blight, again, worldwide distribution, seed propagated. It's mostly from warm and humid areas. There is resistance identified, but mostly outside the species. So breeders have had to make interspecific crosses with 2 other phaseolus species to introduce this resistance. 2 fungal diseases, anthracnose and angular leaf spot. And angular leaf spot is becoming really important, not only in Africa, but also, for example, in Brazil. Again, seed propagated. You have the role of wind and rain in local transmission. They have identified qualitative as well as quantitative resistance. Again, some of these genes have already been tagged. And that was one of the reasons we picked disease resistances, is that we could already start working with existing markers. And then finally here, another fungal disease, but this is, in this case, it's a root rot. And based on information made available by CIAT, we focused on Pythium as the major agent. There are other agents of root rot. And again, there are markers available to select for resistance. So the concept of marker-assisted selection, just let me say a few words, is that instead of selecting directly phenotypically for the gene, this would be a gene for resistance here, breeders actually select for a marker that is physically linked to that gene. Now, why would they want to do so? Well, primarily is because you have increased heritability. If you select for that particular sequence here, which is the marker, it is going to be present all the time. Whereas the resistance, the phenotype for the resistance may or may not be present. The disease may not be present, in which case you cannot select. Okay? So that will lead to an increase in heritability. You can also conduct a selection in earlier generations rather than late generations, because you can select on the basis of individual plants. You can also select at an earlier rather than late developmental stage, for example, before flowering rather than late. And that means that you can select plants and then make crosses with these plants because you have been able to select them before the flowering time. Okay? You can also, and this is important in the cases where we have multiple diseases, you can do concurrent selection rather than consecutive selection. There is only so many diseases you can inflict on a seedling without the seedling perishing, actually. Okay? So the idea is that if you do a selection on the presence or absence of a DNA sequence, you'll be able to advance, okay, concurrently, in parallel. But of course, it needs a final confirmation by phenotypic selection, so selection for the presence of the actual resistance. Okay? So that is what attracts people to actually this concept of marker-assisted selection. Okay? Now, there are conditions to achieve that. You have to establish this linkage between the marker and the gene. So being able to identify that requires also certain techniques. And mainly, it actually requires the knowledge of the sequence, actually. Okay? And that's what has been the role of the project, as I'll show later on, is to make this information available. So let me give you two examples of projects. We have Tanzania at Sokoine University. To give you an illustration, this is a photo of the annual meeting that we hold every year. So what they are focusing on, and what the project is focusing on, is that they have a preferred variety that is preferred by farmers and consumers. And what they are then crossing this variety with sources of resistance. So in this case, MEX54 is a source of resistance to angular leaf spot. Shindi is an earlier variety produced by Sokoine, and it has resistance against the viral disease I presented. And then VAX3 or VAX4 is a common bacterial blight source of resistance. Okay? And so the idea is to develop a version of this Kablanketti variety, but with multiple disease resistances, involving marker-assisted selection. Another project is in Uganda at NACRI. Again, it involves a preferred variety, Nabe 12C, which is a climbing variety. It's a sugar type. Again, you see MEX54 as a source of resistance to angular leaf spot. G2333 is a variety from southern Mexico, a landrace from southern Mexico, and it has 3 different genes for resistance to anthracnose. And then here, Pythium locally is a problem, so you have RWR719, and this is the marker for this resistance to Pythium root rot. Okay? There is also a project of Kenya. Okay? At Davis, what we are doing is, as I told you, is to make available as much as possible this sequence from which we can derive markers. And so one of the ways of doing that is to generate or develop a database called Phaseolus genes, and you can look at it on the web. It has a simple URL right now, address, and it does a number of things. It has a partial sequence of the bean genome. It has a database with all the markers that have been developed or are being developed. It has what is called a genome browser, which is a marker map, but at the scale of the base pairs. Okay? And then it has a comparative map, which is a marker map, but at a genetic scale. So the unit of distance there is the recombination level. It's the centimorgans for the geneticists here. What we also do is to optimize existing markers and develop additional markers. Our experience is that if it's published, you can't just take it for granted. You have to make it work in your own lab. And there are ways of doing that. And then we also had short-term education of African students. And so this is a way of supporting actually the African projects. Some of the concerns I have is that genomics is a moving target. And so what is true in 1 year may not be true the next year in terms of the technologies available. When we started, there were no whole genome sequences available. We used the soybean sequence because soybean is a relative of common bean, separated by 20 million years, but it's still the order of the genes on the chromosomes remains the same in large blocks. And this year, actually today, they have told me that they were going to release the first whole genome sequence for common bean in the US, okay? At the Phytosome website in California. We have, as you see, we have communications between 3 continents, and it's not as good as having direct communication, but we have to make it work. What is the troubleshooting beyond the training periods, the annual meetings and so on, the stays in labs at CIET and at UC Davis? And I think at the beginning of the project, at least, internet access was an issue. It may still be or may not. I think it's improving, but having internet access is really a key for research. So the current status, I think we are halfway towards the development of advanced lines. In terms of bioinformatics, as I told you, we need to integrate this whole genome sequence of common bean into the database, and that is going to facilitate the discovery of markers tremendously. Because instead of having to rely on soybean, we see now that it's much easier to physically map sequences. How do we know, finally, how do we know that we will have been successful? I think clearly the main goal here is the development of advanced lines that not only are preferred by farmers, but that have multiple disease resistances. Okay? I would like to see autonomy in marker-assisted selection of the national programs. People in the ought to be able to develop their own marker-assisted selection programs, as well as autonomy in disease resistance characterization, tagging, and utilization. I think these are the 3. If we can achieve that, I will be very happy, actually. Thanks very much for your attention.